Hello everyone. I'm going to start with the next topic, that is apoptosis. The questions that have been asked so far, they are the write a short note on apoptosis that has been asked as a 5 mark answer, and there is write a short note on mechanism of apoptosis that has also been asked as a 5 mark answer. So let's start with the topic of the day, that is apoptosis. So this is the here is the chart that I have made. It depicts everything where the actual step of the apoptosis happens and how it happens, what type of pathways are involved, or what are the phases that are involved. So let's start with the definition first. What is apoptosis? In simple words, apoptosis is actually ATP mediated programmed cell death. This is the definition that every medical student knows, and they can never ever forget this definition. So, let's start with the definition of proper exact definition of apoptosis. Apoptosis is a type of cell death that is induced by tightly regulated suicide program in which the cells destined to die, they will activate the intrinsic enzymes that degrade the cell's DNA and nuclear and cytoplasmic proteins. In short, it is actually the programmed cell death, which is ATP mediated, and that is induced by the intrinsic enzymes. And these intrinsic enzymes will lead to the death of the degradation of the DNA, cell's DNA, and nuclear and cytoplasmic proteins. So here I'll let you know what are the intrinsic enzymes that are activated and how they are activated. The main thing that we were to learn in apoptosis is what are the caspases and what caspases will lead to the activation of the enzymes. So, starting with the phases of the apoptosis is actually divided into two phases, that is the initiator phase and the execution phase. So, initiator phase is further divided into extrinsic phase, which is also known as cell mediated pathway or the death receptor pathway. Another is the intrinsic phase, which is also known as the mitochondrial pathway. So, starting with the extrinsic phase or the cell mediated pathway or the death receptor pathway, how it happens and what it leads to the activation of which enzymes are activated, that I'll let you know. So, in any stressful condition, what will happen that the monomeric form of the FAS, which is a member of the TNF family, it is actually FAS actually belongs to the TNF family receptors. It is actually present as a monomer form, and whenever there occurs a stressful condition, there this monomer will be converted to the heteromer, and there is increase in the fast ligands which are expressed on the T lymphocytes. This heteromer fast and fast ligand, they will interact with each other, and it will lead to the trimerization of the receptor. This trimerization will further lead to the activation of the FADD, that is also known as the fast associated death domain. This fast associated death domain will lead to the activation of the caspase 8 and 10 from the procaspases 8 and 10. This procaspase 8 and 10 are in they are not activated to the active forms by the flip protein. This flip protein is actually the anti-apoptotic protein. It doesn't lead to the activation of the procaspase 8 and 10 to the caspase 8 and 10. So this is the extrinsic phase or the cell mediated pathway or the death receptor pathway, which leads to the activation of the caspase 8 and 10. Holding this till now, I'll move on to the another phase of the intrinsic of the initiator phase. Extrinsic phase will lead to the activation of caspase 8 and 10, and moving on to the another pathway, that is the intrinsic phase or the mitochondrial pathway. As the name says, mitochondrial pathway, so it would have something to do with the mitochondria also. So, starting with the intrinsic phase. In any stressful condition, like there is withdrawal of the growth factors, or there is DNA damage by the radiations or the toxins or the free radicals, or there is protein misfoldings which lead to increase in the endoplasmic reticulum stress. These stressful conditions, these are these are sensed by the sensors of the cell stress, which are BIM, BID, BAD, NOXA, and PUMA. These are the sensors of the cell stress, and these are BH3 only proteins. They have only one domain in them, that is BH3. These are the sensors of cell stress. These will sense the stress and will lead to increase in the increase in the pro-apoptotic proteins. That is the BAX and BAC. These are the pro-apoptotic protein. These sensors of the cell stress will lead to increase in the pro-apoptotic protein, and they will decrease the anti-apoptotic proteins, that is the BCL2 protein. Okay. Further, this BAX and BAC protein increase in pro-apoptotic protein. These pro-apoptotic protein will form the oligomer, which will insert in the mitochondria. These proteins will form oligomer, and that will get inserted into the mitochondria, forming a pathway through which our two proteins will leak out of the intermitochondrial space, that is the cytochrome C and SMAC and Diablo. These two protein will leak out of the mitochondria. Since cytochrome C will leak out of the mitochondria, this cytochrome C will leak out of the intermembranous space into the cytoplasm, and this cytochrome C will bind with the APAF1. Do remember this protein, that is the APAF1. This cytochrome C will come out into the cytoplasm from the mitochondria, and it will bind with the APAF1. APAF1 is actually the apoptotic activating factor 1. This combination of cytochrome C plus APAF1 is known as apoptosome. This apoptosome will lead to the conversion of procaspase 9 to caspase 9. So I hope I am clear. This this mitochondrial pathway or the intrinsic phase, this leads to the activation of caspase 9. Our extrinsic phase or the cell membrane mediated pathway, this leads to the activation of caspase 8 and 10. Further explaining this thing to you, this SMAC and Diblo, which also leaks out of the mitochondria, this will neutralize the cytoplasmic protein, that is the inhibitor of apoptosis. This will further lead to the apoptosis of the cell. From this initiator phase, we came to know that there is activation of the caspase 8 and 10 and caspase 9. Now, moving on to the execution phase, what these caspases will lead to. Execution phase, since initiator phase, it leads to the activation of caspase 8 and 10 and caspase 9. These caspase 8, 10 and 9 will lead to the activation of caspase 3 and 6. 3 is more important to note, since it will lead to the cleavage of the proteins having the cysteine after the aspartic acid, that will lead to the breakdown of the cytoskeleton. What does it mean? That is any protein that has the cysteine residue, and after the cysteine residue, and any protein having the cysteine residue, and these proteins having the aspartic acid, these caspase 3 will lead to the breakage of those proteins after the aspartic acid. That will lead to the breakdown of the cytoskeleton. This caspase 8 and 10 and caspase 9 will also lead to the activation of the endonuclease. This endonuclease will cause the fragmentation of the DNA, and this fragmentation of the DNA would be at 180 base pair. That will that everything every segment that is after 180 base pair, there would be the breakage. This is known as DNA laddering. This is known as DNA laddering. This cleavage will lead to the chromatin aggregation, and these chromatin will aggregate peripherally in the nucleus. That will lead to the fragmentation of the nucleus. This chromatin condensation happens at the periphery of the nucleus, and it would lead to the nuclear fragmentation. Since I hope I am clear till now that how this happens. There is initiator phase, there is execution phase. Initiator phase will lead to activation of caspase 8 and 10 and caspase 9 via two pathways, and the execution phase with the help of activated caspase 8, 10 and 9 will lead to the activation of caspase 3 and endonuclease. That will lead to the breakdown of the cytoskeleton and the nuclear fragmentation. This will lead to the formation of the apoptotic bodies. What are apoptotic bodies? Apoptotic bodies are actually the membrane bound bodies with or without nucleus. They may have nucleus in them, they might not have nucleus in them. And these apoptotic bodies will send up the eat me signal. These apoptotic bodies will send the eat me signal, and these eat me signal will be recognized by the macrophages, and that will lead to the engulfment of the apoptotic bodies before the breakdown of the membrane, and there will be no inflammation. Now, how the eat me signals are recognized by mitochondria, that I will let you know. Since there is formation of the apoptotic body, these apoptotic bodies, there is phosphatidylserine in the inner side of the membrane. Since this is the inner side of the membrane, there is inner side of since this is inner side of the membrane, and there is a phosphatidylserine over the inner side of the membrane. Whenever there occurs the eat me signal, there will be a flipping of the phosphatidylserine, that is from inside it will move out of the cell, and that will be recognized by our macrophages. And as soon as the macrophage identify the signal, it will immediately cause the breakdown of the membrane, so there is no inflammation. Since there is a thrombospondin also that flips out and is expressed over the outer layer of the membrane, these two, phosphatidylserine and thrombospondin, will form the eat me signal, and this will be recognized by our macrophages, and it will engulf the apoptotic body. This process is known as efferocytosis. This process is known as efferocytosis. I hope I am clear via this chart. What is apoptosis? How it happens? What is the mechanism of it? I hope I am clear to you. Moving on to the main thing that I want you to know, that how to write an answer when the question is asked. I hope I am clear by now what is mechanism of it. Now, how to write it as an answer, I will let you know. Starting with the definition, what is apoptosis? Apoptosis is a type of cell death that is induced by tightly regulated suicide program in which the cells destined to die they will activate the intrinsic enzyme that degrades the cell's DNA and nuclear and cytoplasmic protein. As I have told you the definition earlier also, that is the intrinsic enzymes will get activated. I hope you are clear now which intrinsic enzymes will be activated. That is caspase 3 and endonucleases will be activated, that will cause the degradation of cells DNA and nuclear and cytoplasmic protein. What are the examples where the apoptosis is needed in our body? Even in the physiological processes, we need apoptosis. And what are those processes? First is embryogenesis, the most important process in which we need apoptosis. And another is the organs or the hormone dependent tissues. Um, where the apoptosis is required, and what are those, like in endometrial cell breakdown during menstrual cycle or the ovarian follicle atresia in the menopause. These are the physiological conditions where apoptosis is required. And another example is when we have an inflammation in our body and the cells, they have done their work, like neutrophils, they have done their work, and now they are no longer needed in our body. So there occurs the apoptosis of these cells, like neutrophils after acute inflammation, lymphocytes after the immune reaction. I hope I am clear now. Now, what are the pathological processes that leads to apoptosis? That is DNA damage or the accumulation of the misfolded proteins or infections or the degenerative diseases. These all or the heart diseases. These all are the conditions where the apoptosis is where the apoptosis occurs. um, the other thing that I want you to know is the infections, like HCV infection or HCV infection or sorry for this condition. So, in infections like HCV infection, there is formation of the councilman bodies. That is, a councilman body is also an apoptotic body that is formed in the HCV, and the chemotherapy induced cancer cell death. This condition, there is in this condition there is also apoptosis, or in this condition the cells die, cancer cell die by via both the mechanisms, that is apoptosis also and necrosis also. But the main mechanism that is involved in the chemotherapy induced cancer cell death is the apoptosis. So I hope the examples are clear to you, that in which conditions the apoptosis occur, and in the other condition, in the physiological and pathological conditions in which the apoptosis occur. Moving on to the mechanism of apoptosis, I will be bit short in this because I have earlier explained you in the chart. So, mechanism of apoptosis, there is initiator phase and the execution phase. The initiator phase is further divided into extrinsic phase and the intrinsic phase. So, revising the things with you, that is, extrinsic phase is also known as cell membrane mediated pathway and the death receptor pathway. What it will lead to? It will lead to activation of caspase 8 and caspase 10. And intrinsic phase is also known as the mitochondrial pathway. It will lead to activation of caspase 9. So, how to write if the question is asked, like starting with the initiator phase has the extrinsic phase. Extrinsic phase is also known as cell membrane mediated pathway or the death receptor pathway. How it happens: whenever there occurs the stress to the cell, the fast that belongs to the TNF family receptor, they are monomer in the original form. They will get converted into the heteromer, and there is increased expression of the fast ligands that are expressed on the T lymphocytes. These fast ligands will combine together and will lead to the activation of FADD, that is fast associated death domain. It will lead to the activation of caspase 8 and 10 from procaspase 8 and 10. Procaspase 8 and 10 will be converted to the caspase 8 and 10, which are the active form. This is the extrinsic phase which leads to the activation of caspase 8 and 10. Next phase is the intrinsic phase. Intrinsic phase is also known as the mitochondrial pathway. How this happens: whenever the stressful condition occurs, like growth factor withdrawal or DNA damage like radiation, toxins, free radicals, or the protein misfoldings that leads to endoplasmic reticulum stress, these all these stressful conditions will be recognized by the sensors of cell stress, that is BIM, BID, BAD, NOXA, and PUMA. These sensors of stress, sensors of cell stress will increase the pro-apoptotic enzymes and will decrease the anti-apoptotic proteins. Okay, pro-apoptotic protein, that is BAX and BAC, they will increase, and anti-apoptotic proteins, that is BCL2, that will decrease. Okay, so pro-apoptotic proteins will increase, they will form an oligomer and get inserted into the mitochondrial membrane. That will create a pore or a hole, a kind of hole through which the cytochrome C and SMAC and Diablo will come out of the mitochondria into the cytoplasm. Cytochrome C will leak out of the intermembrane space into the cytoplasm and it will bind with the APAF1. APAF is apoptosis activating factor 1. This combination, cytochrome C and APAF1, is known as apoptosome. And this apoptosome will lead to conversion of procaspase 9 to caspase 9. Another leakage thing, another thing that leaks, another protein that leaks out of the mitochondria, that is SMAC and Diablo. This SMAC and Diablo, it will neutralize the cytoplasmic protein, that is IAPs, that is inhibitors of the apoptosis. So through these two mechanisms, caspase 8 and caspase 10, caspase 9, these are activated. These are the initiator phase. Now moving on to the execution phase. So in execution phase, the caspase 8 and 10, this will lead to activation of caspase 3 and 6. The more important caspase is the caspase 3. There is activation of the caspase 3 that will lead to cleavage of proteins having the cysteine after the aspartic acid. These will cleave the proteins that will have the cysteine and they will cleave the protein after the aspartic acid. This will lead to the breakdown of the cytoskeleton, and it also lead to the activation of the endonuclease. Endonuclease will cause the fragmentation of the DNA and will lead to the chromatin condensation. This chromatin will aggregate peripherally under the nuclear membrane and will lead to the nuclear fragmentation. This leads to the formation of the apoptotic body. There is formation of the apoptotic body, and now these apoptotic bodies will send the eat me signals. These will send the eat me signals to the mitochondria. These apoptotic bodies will send the eat me signals to the mitochondria. Mitochondria will come and engulf these apoptotic bodies. And what these eat me signals are made up of? They are made up of thrombospondin and phosphatidylserine. This thrombospondin will flip out of flip out and will be expressed on the outer layer of the membrane, and phosphatidylserine will be recognized by the macrophages immediately, and they will engulf these um, apoptotic bodies before the breakdown of the cell membrane. Since there is no breakdown of the cell membrane, so no infection occurs. More important to remember, these are the phosphatidylserine and thrombospondin. These two will form the eat me signal. These two will form the eat me signal. So finally, I just want you to remember the very, very important points regarding the apoptosis. What are these points? These are: apoptosis is actually the ATP dependent programmed cell death. What is the marker of apoptosis? Annexin 5. This annexin 5 detects the phosphatidylserine. This is the marker of apoptosis. What if what if we are asked the molecular marker of apoptosis? Obviously, if they are asking us about the molecular means they are asking us about the CD marker. Okay, so molecular marker of apoptosis is CD95 or the FAS. FAS is actually CD95 is the molecular marker of the apoptosis. Hallmark of apoptosis is the nuclear chromatin condensation. Hallmark of neuronal apoptosis is apoptosis inducing factor. Our neurons, they don't have caspase in them. Still they undergo apoptosis. This is because of the AIF, that is the apoptosis inducing factor. Now, how to stain these apoptosis and necrosis? That is via the tunnel staining. Tunnel staining will be positive in case of apoptosis and it would be negative in case of necrosis. So here, this is the final thing that I want you to know, that what are these important points and everything. So this is how you will approach and answer if a question is asked on the apoptosis or the mechanism of apoptosis. Thank you all.